Assalamu alaikum, my dear friends. Myself, M. Rasafanisa from 2nd MS Microbiology. Today I am going to teach you about the glutamic acid production. Now we will see what is glutamic acid. Glutamic acid is a non-essential amino acid that plays a vital role in metabolism, protein synthesis, and neurotransmission. It is also widely used in food, pharmaceutical, and chemical industries. As we all know that from glutamic acid we can get monosodium glutamate, which enhance the food flavor. And microbial fermentation is a primary method for industrial glutamic acid production due to its cost effectiveness, sustainability, and high yield. Now we will see what are the microorganisms used for the glutamic acid production. The primary microorganism which is used for the glutamic acid fermentation is Corynebacterium glutamicum due to its high production efficiency. And other bacteria also we are using, which includes Brevibacterium flavum, Micrococcus glutamicus, Arthrobacter species. These bacteria naturally accumulate glutamic acid under specific fermentation condition. Now about its fermentation process. First, about its inoculum preparation. A pure culture of Corynebacterium glutamicum is grown in a nutrient-rich medium which contains glucose, peptone, yeast extract. The inoculum is incubated under optimal condition, which is 30 to 37 degrees Celsius, pH under 6.8 to 7.2 for 12 to 24 hours. Fermentation medium composition, which includes carbon, nitrogen, mineral salts, biotin, precursors. In carbon source, which includes glucose, molasses, and starch hydrolysate. In nitrogen source, ammonium sulfate, urea, peptones are used. In mineral salts, magnesium sulfate, potassium phosphate are used. Biotin, a growth limiting factor. Limited biotin supply induce the glutamic acid accumulations. In precursors, alpha ketoglutarate acid, which enhance the glutamate synthesis. Regarding the temperature, it should be under 30 to 37 degrees Celsius, and regarding pH, it should be under 6.5 to 7.2, maintained using ammonia or calcium carbonate. Regarding the aeration, high oxygen supply enhance the glutamate production. For agitation, it should be 200 to 500 rpm for even distribution of nutrients and oxygen. Biotin limitation. Excess biotin promotes biomass growth, but limited biotin leads to glutamate excretion. Mechanism of glutamic acid accumulation. In normal conditions, Corynebacterium glutamicum directs glutamate into proteins. When biotin is limited, the cell membrane becomes more permeable, leading to extracellular glutamate secretion. Excess ammonia ions or surfactants like 2-in-80 also enhance the glutamate leakage. Recovery and purification of glutamic acid. Cell removal. The fermented broth is filtered or centrifuged to remove the bacterial cells. The first step, which we will use the filtration or centrifugation method to remove the bacterial cells. In precipitation, the glutamic acid is precipitated by adjusting the pH to 3.2 isoelectric, which is the isoelectric point. Crystallization, the precipitate is crystallized and dried. In refining, the further purification is done through the ion exchange chromatography or solvent extraction. The final product, the purified glutamic acid, is dried and packed for commercial use. This is the flowchart for the production of glutamic acid. Let's see what are the factors affecting the glutamic acid production, which includes substrate concentration. High glucose level enhance yield. Oxygen supply, higher aeration favor glutamate production. pH control, optimum pH prevent by product formation. Mutation and genetic engineering, enhancing the metabolic pathway, improve production. I hope you have well understood regarding the glutamic acid production. Thanks for watching.